Welcome class. In this session, we will be discussing the further steps of aerobic respiration. Among that, the one is Krebs cycle. So this Krebs cycle is included under aerobic respiration because, so here, the particular reaction or particular cycle, it will be occurring in case of the mitochondria. So followed by the glycolysis, both Krebs cycle and electron transport system, they occur in a mitochondria, which is known as the powerhouse of the cell. And in case of Krebs cycle, it is also called as TCA cycle, or it is called as citric acid cycle. So it is called TCA, where TCA refers to tricarboxylic acid cycle. So TCA is tricarboxylic acid, or it is also called as citric acid cycle. So that is because, so here the first stable product formed during this particular reaction will be a tricarboxylic acid named as citric acid. So that's why the alternate name is given for the Krebs cycle. And this Krebs cycle has been briefly explained by the Hans Krebs. Sir Hans Krebs was the one who elucidated the detailed steps of the Krebs cycle. So hence it is also called as Krebs cycle. And the site of reaction will be the mitochondrial matrix. So in the mitochondria, it will be having a double membrane structure within which there will be a matrix. So the mitochondrial matrix will be containing enzymes that are essential for this Krebs cycle. And here, during the Krebs cycle, so here, it requires the product of glycolysis. That is, it requires pyruvic acid for the continuation of the Krebs cycle. So this pyruvic acid is produced in the cell cytoplasm, which is the product of glycolysis. And this pyruvic acid, it has to be transported into the mitochondria. And for that, this pyruvic acid, it undergoes certain chemical changes within it, and altogether it is called as preparatory step. And what happens in this preparatory step, we'll see. So here it is also called as pyruvate oxidation. So in this step, the pyruvic acid that is present in the cell cytoplasm, so it will undergo decarboxylation as well as dehydrogenation. That is, a molecule of carbon dioxide will be removed, and then here the NADH, it will be, NADH and H plus will be formed upon reduction of the NAD plus. Okay, so hydrogen ions will be accepted from the pyruvic acid and the carbon dioxide will be removed from the pyruvic acid, and this particular reaction, it will be catalyzed by the enzyme pyruvate dehydrogenase. So pyruvate dehydrogenase catalyzes this reaction, and in addition to this, so this pyruvic acid, upon undergoing decarboxylation and dehydrogenation, so it will bind with it of the coenzyme A. So that's coenzyme A is a sulfur containing molecule. So it is a sulfur containing coenzyme which combines with that, and it results in the formation of a product that is called as acetyl coenzyme A. So this results in the formation of acetyl coenzyme A. So here, for 2 molecules of pyruvic acid produced in glycolysis, 2 molecules of acetyl coenzyme A will be formed. And this acetyl coenzyme A, now it is permeable through the mitochondrial membrane. So hence, from the cytosol, so this acetyl coenzyme A will be transported into mitochondrial matrix, where the further steps of the Krebs cycle will proceed. So now we'll look into the steps of Krebs cycle in detail. So here the preparatory step is mentioned. So that is prior to the Krebs cycle. So this is the preparatory step. So here pyruvic acid, it combines with, it undergoes decarboxylation and dehydrogenation and combines with coenzyme A, resulting in the formation of acetyl coenzyme A. So this acetyl coenzyme A is a 2-carbon compound. So now this 2-carbon compound, that is acetyl coenzyme A, it combines with the 4-carbon organic acid called as oxaloacetic acid, and results in the formation of a 6-carbon organic acid that is called as citric acid. And this process, it occurs in the presence of water medium, and the process, this particular step, the first step is called as condensation here. This step is called as condensation, where two molecules, they are condensed to form a one larger molecule. So 2-carbon compound and 4-carbon compound, they are condensed together in order to form a 6-carbon compound that is called as citric acid, and the reaction is catalyzed by the enzyme citrate synthase. So next, once the citric acid is formed, so now it undergoes the second step that is called as dehydration. So in case of dehydration, a molecule of water will be removed from the citric acid, okay, and it results in the formation of one more 6-carbon acid that is called as cis-aconitic acid, or it is called as cis-aconitate, and the reaction is catalyzed by the enzyme aconitase. And in the third step, again the removed water molecule will be added back to the cis-aconitate, but water will be added into the different carbon of this aconitate. Generally here, when water is removed, it will be removed from the second carbon atom and of the citric acid to form cis-aconitate. So for the cis-aconitate to the third carbon atom, a molecule of water will be added, and thus it results in the formation of isomer of citric acid that is called as isocitric acid. And both the reaction here, that is, which is coupled with formation of cis-aconitate and isocitrate, are catalyzed by the same enzyme that is called as aconitase. So both the reactions are catalyzed by the same enzyme. So now, once this isocitrate is formed, so this isocitrate, it undergoes dehydrogenation. So the fourth step is dehydrogenation. So it undergoes dehydrogenation, resulting in the formation of one more, so isocitrate is also 6-carbon acid. It results in the formation of one more 6-carbon acid here. So that acid is not mentioned. So that is called as, so isocitrate, first it undergoes dehydrogenation, resulting in the formation, I'll just mention here, of an organic acid that is called as oxalosuccinic acid. So it results in the formation of organosuccinic acid, which is again a 6-carbon acid. So followed by this dehydrogenation, so now this oxalosuccinic acid, it undergoes decarboxylation. So fifth step will be decarboxylation here. So it undergoes decarboxylation and it loses a molecule of carbon dioxide. As a result, it results in the formation of a 5-carbon organic acid or a keto acid that is called as alpha ketoglutaric acid. And here, during the dehydrogenation, the hydrogen ions donated by the isocitric acid will be accepted by the NAD plus and it gets reduced into NADH and H plus. And here, both the formation of oxalosuccinic acid and alpha ketoglutaric acid. So it is catalyzed by the same enzyme that is called as isocitrate dehydrogenase. So this enzyme catalyzes the conversion of isocitric acid into ultimately a 5-carbon keto acid that is called as alpha ketoglutaric acid. So once this alpha ketoglutaric acid is formed, so now this alpha ketoglutaric acid, it undergoes dehydrogenation as well as decarboxylation together. So here, sixth step, it involves both dehydrogenation as well as decarboxylation. So as a result, a molecule, hydrogen ions are also removed and a carbon dioxide is also removed. The removed hydrogen ions will be accepted by the NAD plus to form NADH and H plus, and the carbon dioxide will be released. And this carbon dioxide release is coupled with the addition of coenzyme A group here. As a result, it results in the formation of a high energy compound which is a 4-carbon compound that is called as succinyl coenzyme A. And this succinyl coenzyme A formation from the alpha ketoglutarate, it is catalyzed by the enzyme called as alpha ketoglutarate dehydrogenase. So this particular enzyme catalyzes this reaction. And once the succinyl coenzyme A is formed, so as I said, it is a high energy compound. So here, this succinyl coenzyme A, in the presence of water molecule, so it gives up its coenzyme group and it gets converted into a 4-carbon organic acid that is called as succinic acid. And this step, it is called as synthesis. So that is because here synthesis of energy rich compound takes place here. So what is that we'll see. So succinyl coenzyme A, it gives away its coenzyme A group, resulting in the formation of succinate or the succinic acid. And this succinic acid formation is catalyzed by the enzyme succinyl coenzyme A synthetase. And here, as coenzyme A is removed from the succinyl coenzyme A, the energy released during this particular process is sufficient enough to add a phosphate to the guanosine diphosphate, resulting in the formation of guanosine triphosphate. And this guanosine triphosphate, it can directly form the adenosine triphosphate or the ATP later in the mitochondrial matrix. So that's why this particular step is coupled with the synthesis of energy-rich compound. So it is called as synthesis. So once the succinate is formed, so the 8th step here is again the dehydrogenation. So in case of dehydrogenation, so now the succinic acid will be converted into fumaric acid, where succinic acid, it loses its hydrogen ion and it gets converted into fumaric acid, and the hydrogen ions removed from the succinic acid will be accepted by the reducing agent, that is not NAD here, it is one more reducing agent that is called as FAD, where FAD refers to flavin, adenine, dinucleotide. So it is a type of a flavoprotein or a coenzyme, so which will be accepting this hydrogen. And this particular reaction is catalyzed by the enzyme succinate dehydrogenase. And one more importance of this step here is, so this succinate dehydrogenase enzyme, so it will be near to the inner mitochondrial membrane. So even though reaction is occurring in the mitochondrial matrix, so this succinate dehydrogenase, it will be associated with the inner mitochondrial membrane, and this particular enzyme, it is also coupled with the electron transport chain, that is the next step of the aerobic respiration. So hence the succinate dehydrogenase is the enzyme which links the Krebs cycle to that of the electron transport mechanism. So that is the importance of the succinate dehydrogenase enzyme. And the reducing agent used here will be FAD, which will be reduced into FADH2. And once the fumarate is formed, so the third step is, the next is the 9th step. So the 9th step is called as hydration. So where a water molecule will be added to this fumarate in order to form a one more 4-carbon acid that is called as malate or the malic acid. And the reaction will be catalyzed by the enzyme fumarase. So then once this malic acid is formed, now this malic acid, it undergoes dehydrogenation. So that is the 10th step. So it undergoes dehydrogenation and it results in the formation of the or regeneration of the oxaloacetic acid. And as a result of dehydrogenation, the hydrogen ions removed from the malic acid will be accepted by the NAD plus to form NADH plus H plus, and the reaction will be catalyzed by the enzyme malate dehydrogenase. So that is regarding the detailed account of citric acid cycle or the Krebs cycle. And here this Krebs cycle is also important in case of biosynthetic process also. That is because the intermediates produced in the Krebs cycle, they are coupled with the synthesis of the proteins. So, or the amino acids, they are involved in the amino acid biosynthesis, such as, so the alpha ketoglutarate, which is formed in the Krebs cycle. So it is coupled with the synthesis of amino acid, such as glutamic acid, aspartic acid and all, whereas pyruvic acid, so it is coupled with the synthesis of amino acid called as alanine. Similarly, the succinate, which is formed, so it is used in the production of the tryptophan, that is one more amino acid. So this tryptophan is used in the biosynthesis of indole acetic acid or the natural auxin. Similarly, this amino acids such as glutamic acid, alanine, aspartic acid, all these are used in the protein biosynthesis. So hence the intermediates that are produced in the Krebs cycle, they are used in the biosynthesis of proteins, nucleotides, as well as certain plant hormones. So that is regarding the importance of Krebs cycle. And if we look in case of the end products of Krebs cycle, so end products include, so this reaction is in terms of one molecule of pyruvic acid. So as here, 2 molecules of pyruvic acid is formed as a result of glycolysis. All the products that are formed per cycle, it has to be calculated twice here, because 2 times Krebs cycle will be operating for 1 molecule of glucose. So end products per glucose molecule if we consider. So in examination, the question can be asked with reference to per pyruvic acid or per glucose. You have to read the question properly and you have to answer. So we'll consider the end product per glucose molecule here. So per glucose molecule, we are getting, in the preparatory step, we will get 2 molecules of carbon dioxide. So this is in the preparatory step. We'll have 2 molecules of carbon dioxide. So here, pyruvate, when it is decarboxylated into acetyl coenzyme A, 1 molecule of carbon dioxide is used, as 2 molecules of pyruvic acids are involved. So hence 2 molecules of carbon dioxide will be formed. For 1 pyruvic acid, 1 NADH molecule is formed. So for 2 pyruvic acid molecule, 2 NADH plus H plus. So this will be the end products of the preparatory step. And coming to the end products of Krebs cycle. So in case of Krebs cycle, if you see, so totally we'll get 1, 2. So this is 1, then 2, and 3 molecules of NADH for 1 pyruvic acid. So as we are calculating per glucose molecule, so we'll get 6 molecules of NADH plus H plus molecules. Similarly, so 1 FADH2 is formed here for 1 pyruvic acid. So for per glucose, we'll get 2 molecules of FADH2. And then, so here 1 molecule of ATP will be directly synthesized per pyruvate. So for per glucose, we'll get 2 molecules of ATP. So these are the end products of the Krebs cycle. So these end products will be utilized in the electron transport chain in order to release the energy. So as per the equation of the respiration, so it releases 6 molecules of carbon dioxide, right? So here 2 molecules are released. Similarly, here 2 molecules of carbon dioxide are released totally. So in the Krebs cycle, we'll get 2 molecules of carbon dioxide in the preparatory step. 4 molecules of carbon dioxide will be released in the Krebs cycle. So totally 6 molecules of carbon dioxide as per reaction. Equation of the respiration will be released by the end of the Krebs cycle. So release of water molecule and the energy. So that can be seen in case of the next step, that is electron transport chain or the electron transport mechanism, that we are going to discuss in the next session.